Good morning. Today I will discuss one of the very important biomolecules, nucleotides and nucleic acids. Other biomolecules are proteins, carbohydrates, and lipids. These biomolecules are important building blocks of life and they perform important functions in organisms. Nucleic acids are the polymers of nucleotides and they constitute 5 to 10% of the cell. So the nucleotides are the constituents of nucleic acids. Nucleic acids are of two types, DNA and RNA, and they store genetic information and transfer this genetic information to the daughter cells during cell division. Nucleic acids also transfer information to mRNA and to proteins. Nucleotides such as ATP and GTP are forms of energy. ATP is the primary carrier of energy in cells and provides energy for various processes. GTP energy is used for protein synthesis and gluconeogenesis. GTP is also essential for G protein coupled signal transduction pathways during cell signaling. Cyclic AMP is also a second messenger and it is involved in cell signaling. The nucleotides, which are the building blocks of nucleic acids, are made up of three components. A nitrogenous base, which may be a purine or a pyrimidine, pentose sugar, which may be a ribose sugar or a deoxyribose sugar, and 1, 2, or 3 phosphate groups. The purine bases are of two types, adenine and guanine, and both adenine and guanine are found in DNA as well as in RNA. Pyrimidines, thymine is found in DNA, cytosine is found both in DNA and RNA, and uracil is found only in RNA. The sugars are of two types, ribose sugar and 2-deoxyribose sugar. These are pentose sugar made up of 5 carbons. Ribose, pentose sugar is found only in RNA and 2-deoxyribose sugar, which is also a pentose sugar, is found in DNA. The basic difference between ribose and 2-deoxyribose sugar is that in ribose sugar, at 2' carbon, hydroxyl or OH group is present, which is absent in 2-deoxyribose sugar. And the presence of hydroxyl group at 2' carbon in RNA makes it a very unstable molecule in alkaline conditions. The nucleosides are made up of a base and a sugar. So base plus sugar is a nucleoside. And if one phosphate group attaches at the 5' position of the pentose sugar in a nucleoside, then nucleotide monophosphates are formed. If two phosphates attach, nucleotide diphosphate is formed, and if 3 phosphate groups are present, then nucleotide triphosphate is formed. For example, in the diagram, adenine is a nitrogenous base. It attaches to the C1 carbon of the pentose sugar, forming a nucleoside. Phosphate group attaches at the C5 position, forming a adenosine 5 monophosphate. The second phosphate group attaches to form adenosine 5 diphosphate, and the third phosphate group attaches to form adenosine 5 triphosphate. The two types of nucleic acids are DNA and RNA, and the difference between DNA and RNA is in DNA, the pentose sugar is 2-deoxyribose, in which hydroxyl group is not present at 2' carbon position, so DNA is a more stable structure. In RNA, the pentose sugar is ribose sugar, in which at 2' carbon hydroxyl group is present. DNA contains 4 types of nitrogenous bases, adenine, guanine, cytosine, and thymine. RNA also contains 4 types of bases, adenine, guanine, cytosine, and uracil in place of thymine. RNA also contain some modified bases such as dihydrouridine or pseudouridine, which are present in transfer RNA. DNA is found predominantly in the nucleus. It stores and transfers genetic information. DNA is also found in mitochondria and chloroplast. RNA is found throughout the cell and it has different forms such as mRNA, transfer RNA, and ribosomal RNA. mRNA, transfer RNA, and ribosomal RNA are involved in protein synthesis. mRNA is also known as coding RNA. Transfer RNA acts as an adapter molecule and it brings the amino acid during protein synthesis, and ribosomal RNA form the ribosomes. DNA mostly occurs in the double-stranded form, whereas RNA may be single-stranded or double-stranded or can be present in RNA-DNA duplex form. The nucleotides are joined together by phosphodiester bonds to form the nucleic acids. During the formation of phosphodiester bond, the 5' phosphate group of one nucleotide unit is joined to the 3' hydroxyl group of the first nucleotide. So always a nucleotide attaches at the 3' position of the next nucleotide. The covalent backbones of nucleic acids are made up of phosphate and sugar residues. Nitrogenous bases form the side groups. By convention, nucleic acids or oligonucleotides are always written in 5' to 3' direction. 5' end is written at the left side and 3' end is written at the right side. During the formation of phosphodiester bond, the phosphate group, which is present at the 5' position of the carbon, forms a phosphodiester bond with the 3' OH group of the first nucleotide. This is a condensation reaction in which a molecule of H2O is released and two phosphate groups are released in the form of pyrophosphate. So in DNA and RNA, nucleotide monophosphates are present. And the polarity of DNA and RNA is 5' to 3'. Always a nucleotide attaches at the 3' end and chain grows from 5' to 3' direction. So the phosphodiester bonds are the bonds which help in joining the two nucleotides together. The formation of phosphodiester bond is a condensation reaction in which a water molecule is released and a pyrophosphate is also released. Hydroxyl group, which is present in the ribose sugar at 2' position in RNA, can take part in a cleavage reaction which can cleave the phosphodiester bond, especially during alkaline conditions. So RNA is a very unstable molecule and it can degrade in the cell within 30 minutes. DNA, which does not have hydroxyl group at 2' carbon position in the pentose sugar, is a stable molecule. It exists during our lifetime. It acts as a repository of genetic information, and from millions of years old sample, DNA can be extracted. So in this diagram, the instability of phosphodiester bond is shown. Since RNA contain ribose sugar which has hydroxyl group at 2' position, this hydroxyl group acts as a nucleophile in an intramolecular displacement, especially under alkaline conditions, thereby breaking the phosphodiester bond. DNA which lacks 2' hydroxyls is stable under similar conditions. In a double-stranded DNA, hydrogen bonds are formed between the two strands. The pairing is very specific. A always bonds with T and G always bonds with C. Thymine or uracil have one hydrogen bond donor and one hydrogen bond acceptor. Adenine also has one hydrogen bond donor and one hydrogen bond acceptor. Thymine is always hydrogen bonded with adenine in DNA with the help of two hydrogen bonds. Cytosine has one hydrogen bond donor and two hydrogen bond acceptor. Guanine has two hydrogen bond donor and one hydrogen bond acceptor. So in DNA, cytosine always pairs with guanine and it forms three hydrogen bonds. Chargaff studied the cellular DNA of various species and he concluded that the adenosine residues are always equal in number to thymidine residues and the number of guanosine residues was always equal to the number of cytidine residues. That means A is equal to T and G is equal to C. Sum of purine residues equals the sum of pyrimidine residues. A plus G is equal to T plus C, or purines are equal in number to pyrimidines. So the ratio is A plus G upon T plus C is always unity or 1. Chargaff studied the percentage of A, T and G and C in viruses, bacteria, fungi and eukaryotes like humans and found that the percentage of A was always equal to the percentage of T and percentage of G was always equal to percentage of C. So according to Chargaff's rule, A plus G is equal to T plus C, or purines are always equal to pyrimidines. Rosalind Franklin and Maurice Wilkins studied the X-ray diffraction pattern of DNA, and from their study they proved that DNA is a double helix with two periodicities along their long axis. The primary periodicity was 3.4 angstrom and the secondary one was 34 angstrom. This was the X-ray diffraction pattern obtained by Rosalind Franklin and Maurice Wilkins. This pattern shows that DNA is a double helix. Watson and Crick, from the available data of Chargaff and Franklin and Wilkins, postulated the three-dimensional model of DNA. According to Watson and Crick model of DNA, DNA is a double helical structure. The two helix or two strands are antiparallel. One has a polarity of 5' to 3' and the other one has a polarity of 3' to 5'. Sugar and phosphate make the backbone and nitrogenous bases are present as a side group. In the two strands, A always pairs with thymine and G always pairs with C. The minor groove and major groove are present in DNA double helix. Distance between two nitrogenous bases is 3.4 angstrom and one complete turn has a length of 34 angstrom. In one turn, Watson and Crick said that 10 base pairs are present, which was later revised to 10.5 base pairs, and the length of one turn was 36 angstrom. So the two DNA strands, according to Watson and Crick model, are complementary and antiparallel. This three-dimensional model of Watson and Crick was able to explain the synthesis of a new strand of DNA. Since the nucleotides are joined together by base pairing rules, so one strand of DNA acts as a template and a new strand is synthesized on the older strand. The replication of DNA or the synthesis of DNA is said to be semi-conservative. During the replication of DNA, the two strands of DNA separate. They act as a template for the formation of a new strand. New strand is always synthesized in 5' to 3' direction. Watson and Crick gave the structure of DNA and it is known as bDNA or B form of DNA, which is the most stable structure under physiological conditions. Two other structural variants of DNA are also known, which are known as A form and Z form of DNA. The difference between A, B and Z form of DNA is that A and B form of DNA are right-handed helix and Z form of DNA is left-handed. The diameter of A form of DNA is 26 angstrom, B form of DNA has a diameter of 20 angstrom and Z form of DNA has a diameter of 18 angstrom. There are 11 base pairs per helical turn in A form, 10.5 base pairs per helical turn in B form, and 12 base pairs in Z form. The helix rise per base pair is 2.6 angstrom in A form, 3.4 angstrom in B form, and 3.7 angstrom in Z form. B form of the DNA is known as Watson and Crick DNA. It is the most stable form of DNA under physiological conditions. When we treat DNA with extremely high temperatures or change in pH, it causes denaturation or melting of double helical DNA. Denaturation or melting means the hydrogen bonds between the two strands of DNA are disrupted, and this causes unwinding of the double helix to form two single strands. During denaturation or melting, no covalent bond in the DNA is broken. If we bring the temperature or a pH to the normal conditions, two strands of DNA again form a double helical structure. So it is known as renaturation of DNA. When the two strands of DNA are present, it absorbs less UV light. This is known as hypochromic effect. The denaturation of double-stranded nucleic acid results in increase in absorption of UV light, and it is called hyperchromic effect. The temperature of denaturation or melting point temperature depends on GC content of the DNA. If a DNA has a higher GC content, its melting point is higher because G and C are base paired with the help of 3 hydrogen bonds. So if a DNA molecule is a longer structure, then it has a higher melting temperature, and if it has a higher GC content, the melting temperature is higher.